The first person I want to announce is Nick Radar. He's been a long MMS member. He's done a lot of, he's done gone on the NAMA foray as a Merrick Scholarship winner. Nick Rader is a PhD candidate in the Department of Plant Pathology at the University of Minnesota, Twin Cities. He's a mycologist and forest pathologist who's advised by Professor Robert Blanchette, who I see on the meeting tonight. Thank you for joining us. Nick has studied fungi associated with the emerald ash borer. I'm focusing on canker-causing fungi associated with the beetle and their potential to contribute to the mortality of ash trees. His current work is on the biosurveillance of invasive forest pathogens. So I want to thank him so much for being here tonight and sharing all he has learned over the years. And yeah, we're really excited to have him as our graduate scholarship winner this year.
Yes, thank you, Kathy, and thank you everybody for your attention this evening and good evening. So my presentation this evening is going to be on the biosurveillance of invasive forest pathogens in Minnesota, and my contact information, my email is at the bottom there if you want to contact me, and I'll kind of have a call to action at the end of this, and I'll put my email at the end of this presentation as well. So if you don't jot it down now, feel free to jot it down at the end. Oops. So a little bit about me. Kathy did a very good job introducing me. I'm just going to reiterate it. So I received my Bachelor's of Science in Plant Science from the University of Minnesota in 2018. I then promptly started my Master's of Science in Plant Pathology at the University of Minnesota the same year. And I studied the fungi associated with the emerald ash borer. And like Kathy mentioned, it was the canker-causing fungi and their potential to contribute to ash tree mortality. And I think Colin is going to talk a little bit about that research when he talks about the emerald ash borer later this evening. I then transitioned into a PhD program in 2020, and I'm actually finishing up next week. I defend my dissertation, and the title of my dissertation is the same as the presentation here, the biosurveillance of invasive forest pathogens in Minnesota. And like Kathy also mentioned, I'm advised by Dr. Robert Blanchette, the gentleman in the corner, your left-hand corner of your screen here. So the outline for this evening, I'm just briefly going to discuss historically important invasive forest pathogens, and then I'm going to go into my research on biosurveillance of invasive forest pathogens, and then just briefly touch on the future of biosurveillance in Minnesota. So historically invasive forest pathogens. I show this picture a lot. This is people in the Appalachian region standing next to a big chestnut tree. But before we talk about the pathogens, I want to talk about the live imports in the United States. And you don't have to get too bogged down on the pictures here. I just want to show this picture, and I show this picture in a lot of my presentations, just to reiterate how much plant materials actually come into the United States per day. You can see the D picture there. There's just boxes and boxes and masses of plant pathogens, not plant pathogens, but plants coming in, and that's a potential risk for introduction of plant pathogens in the United States and also Minnesota. So the first pathogen I want to talk about, I'm sure most of the people on this call are familiar with, is chestnut blight caused by the fungus Cryphonectria parasitica. And this is a canker fungus that destroyed, functionally made the chestnut tree or the American chestnut tree extinct. And chestnut trees were very large parts of the people of the Appalachian Mountains and the Smoky Mountain regions. And up the East Coast, the nuts were used as feed for cattle, also feed for humans. And the wood is very resistant to decay. You see people craft a lot of nice wood fixtures out of it and furniture. So it was really sad that this pathogen was introduced to the United States and killed all these chestnut trees. The next pathogen I want to talk about, probably a little closer to home, I'm sure people are familiar with elm trees, is Dutch elm disease, caused by the fungus Ophiostoma nova almi or Ophiostoma almi. And Ophiostoma almi was came in or introduced in the United States via veneer from the Netherlands in 1928. The fungus was then reintroduced to Europe and came back and was reintroduced to the United States in 1940, and it's had staggering impacts in Minnesota. There's pictures out there of boulevards lined with elm trees, and elm trees have that nice cathedral-like structure, and they've all, or most of them, have succumbed to Ophiostoma nova almi or Dutch elm disease. The fungus is vectored by a beetle in this case. So you have the native elm bark beetle on the left-hand side of your screen here, Hylogorophoros rufipes, and then you have two bark beetles, One's the European bark beetle, Scolytus multistriatus, and then one from Russia, Far East. So those are historically invasive and important pathogens we have to keep in mind when we're doing our research, because we don't want to have another Dutch elm disease or another chestnut blight on our hands. We want to be able to mitigate these pathogens before they can cause large amounts of losses. So these are some of the pathogens and diseases that threaten Minnesota. These aren't present, but we're actively looking to make sure they're not here. So the first one is larch canker. And then the second one is ash dieback. But this is a big problem in Europe, but who knows how big of a problem it's going to be here now with the emerald ash borer killing most of our trees, because all of our ash trees are susceptible to emerald ash borer. Next one is thousand cankers disease, pitch canker. And two, we're going to take a closer look at this evening, and what my research has been on is Heterobasidion and Phytophthora. So before I begin and start talking about Heterobasidion, I just want to preface this with why is this research needed and why it's important. And it's important to survey and assess what potentially invasive pathogens are in the forest, so we know what's there. And we can also protect the forest, either natural, state, or urban parks of these potentially invasive forest pathogens. And we want to really mitigate the large economic losses. So you want to catch these, and we want to catch these pathogens before they can establish and have another chestnut blight or Dutch elm disease on our hands. And we also want to preserve the forest for future generations. I'm sure almost everyone, if not everyone on this call, really likes going and enjoying state parks and looking for mushrooms at state parks or even on private land. And that's what we're trying to preserve. We're trying to keep healthy trees for the next generation and the next generation. So the first pathogen, Heterobasidion root disease, I'll sometimes refer to it as HRD. So if you see the acronym HRD, it means Heterobasidion root disease, the same thing here. And it's caused by the fungus Heterobasidion irregulare. And it was formerly known as Heterobasidium annosum and annosum root rot, but it's since been parsed out with genomics in a Heterobasidion occidentale, which occurs mostly on the west coast up to the Rocky Mountain regions. And then you have Phytophthora irregulare, which occurs on the east coast. And I'll show some maps about where it occurs. And it's slowly, or not slowly, it's rapidly coming into the Midwest. And it's found in most of the Midwest states. Its main host is red pine, and that's significant being here in Minnesota because red pines are a state tree. And it can infect other conifers like white pine and fir trees. And it was actually found in Minnesota in 2014 in Winona County and eradicated. And I'll get into that a little more in a couple of slides. So the distribution of Heterobasidion irregulare, the fungus that causes HRD or Heterobasidion root disease, is seen and depicted here. So you can see it's mostly on the East Coast, but it has since moved into the Midwest. So Michigan has Heterobasidion, as well as Wisconsin and some places in Iowa. And the red just indicates the disease has been confirmed there. And then kind of like the orange or the cream is the disease has been reported or is expected, but has not been confirmed either by the Department of Natural Resources or the USDA or the MDA. And let's just zoom in a little bit on the Minnesota and Wisconsin, because these are two key players in my research. So Wisconsin, you can see the red counties there. That's where it's been reported and confirmed. And then you can see Minnesota here. This is Winona County. This is where it has been reported and confirmed, and it has been eradicated. So the first location of Heterobasidion root disease or HRD was in Miniska, kind of up down Highway 61 by Weaver, Minnesota, in the Winona County. And it was eradicated by the Department of Natural Resources. Basically what they did is they cut down all the trees in this forest stand where they found it, and then they pulled up all the stumps so it can spread from tree to tree. And that's significant with this pathogen because it can spread underground. So these are what the fruiting bodies look like. They are basidiocarps. So this is a basidiomycete fungus, and they occur around the base of the conifer trees that infects mainly red pines. And they often can be quite hard to find because of the duff layer. The one on the left-hand side of your screen is very easy to see, but it's a little more tricky with the duff, and I've circled it there for you. So this is a closer look at the fungus, a little closer up. And I want to encourage everybody on the call, and if you're seeing this, if you're watching this recording, if you see fruiting bodies like this, to let me know. Either email me or email the Department of Natural Resources, and if you see this in conifer stands, because it's a really significant pathogen we don't want here. So, and then the right-hand side of your screen here is the popcorn stage. So this is when the fungus is immature. It kind of looks like popcorn, and we give it the popcorn name stage. And the reason the fungus is called Heterobasidion irregulare is because of the irregular pores you can see here, and you can identify it based on the irregular pores, and there's a sterile margin on the outside of the fungus. So a little bit about the disease cycle. So what happens is in places where this disease is prevalent, like say in Wisconsin, pine plantations that are either thinned mechanically or by just cutting down a tree with your chainsaw, if the spores are in the area, the spores will land on the stump. The spores will then penetrate the stump and then spread from tree to tree underground. And once this fungus actually gets into a forest stand, it's almost entirely impossible to eradicate because it spreads underground and causes large amounts of losses in the conifer trees and makes these what we call circles of death. It also can come in on wounds. So it doesn't have to come in on stumps. It can also come in if you're thinning your plantation and the fungus is around, it can get in through the wounds and functionally do the same thing, get into the root system and spread from tree to tree. So the situation in Wisconsin. So Heterobasidion was actually discovered in Adams County in 1993, this county here, and action was not promptly taken. So Wisconsin really didn't monitor for the pathogen, didn't really care about the pathogen. And as of this year, there have been 30 counties that have reported confirmed HRD. And 4 of those counties you can see here are adjacent to Minnesota, which leads to a problem. We don't want this fungus here in Minnesota. So what can we do about it? Before we talk about that, we'll talk a little bit about the management. So how you can manage this if you're a forester in these locations, say in Wisconsin where this fungus is found, you can actually spray your stumps with borax, and that'll prevent the spores from penetrating the stump and getting into the root system. And you can also use fungus as a biocontrol, Flebia gigantea. So there's just 2 of the management aspects of it. Oops. So what does my research do and what does my research entail? So I'm going to talk a little bit about the spore collection and the spore traps that I use to detect and trap these spores. So the first spore sampler here is the Burkhardt air sampler, and it's a cyclone air sampler. So it's got a fin on the back of it, and it runs on battery power. And what it essentially does is it sucks the spores and the air in from wherever you place it. And the spores are actually, they come through the apparatus part here, and they're deposited into a microcentrifuge tube, you can see here. And then everything heavy is pulled down, like the spores, pollen, insects, and everything that isn't heavy, like the air, is actually shot out the top. So these collection tubes get pretty full of pollen, insects, and even spores when I'm out there sampling. Another sampling method is a sampling air sampler is a rotor rod air sampler. And this is an apparatus that it's got these arms that come down, and on these arms are these plastic rods, and they're greased with silicone. And what these do is they're impact rods. So this spore trap spins really fast, and then the arms come down, and what they do is they impact everything in the environment. So they come down and they sample the environment, and then they go back up into the little spore trap here every hour. And you can set these to sample for as little or as long as you want. So I set mine to sample for like 6 minutes every hour, just to get an idea of what's around in the environment and if Heterobasidion is there. And the last spore sampler, and this one is really cheap and really easy to deploy, is an airport windsock affixed with 2 greased microscope slides like you see here. And this is a cheap method to spore trap because you can place it pretty much anywhere. It doesn't run on any batteries, and it moves with the wind, which is really nice for catching the spores. So this is just a quick pros and cons, and I'll let you just look through this quickly. And yeah, so each device has pros and cons, and depending on what you want to get out of sampling in your stand or how sensitive you want your sampling to be, you would pick one of these, each of these spore samplers. I should note, or I should have you note that both of these run on battery power. So you don't really want to carry these into the forest very deep because you have to take a car battery with you and change the car battery out every week for the Burkhardt and every 3 to 4 weeks for the rotor rod spore sampler. So that's also something to keep in mind when you're sampling or when I'm sampling. So this is just a little bit of my sampling scheme. So I set up these spore traps on the Minnesota-Wisconsin border. So you can see Minnesota here on the left-hand side of your screen and Wisconsin on the right-hand side of your screen. And what I do is I deploy the spore samplers all along Highway 61 because we want to be able to catch the spores if they're coming over from Wisconsin. We also do spore trap at a couple places in Wisconsin. So this is Perot State Park, and then this is a Burr Oak, Wisconsin, and this is on private land. And basically we use these as positive controls for the first part of my research, just to make sure that the spore samplers were working like we said they were, and then to compare the spore samplers side by side. So this is just a list of my Wisconsin sites. So yeah, Burr Oak, our positive control, Perot State Park, and Ceylon State Park. And if you're not familiar with where Ceylon State Wildlife Park is, it's pretty much over the Wisconsin border, right across from William O'Brien. And then these are my Minnesota sites. So I sample at Weaver, Minnesota, Whitewater, Miniska, Lake City DNR office, William O'Brien State Park, Itasca State Park, and Great River Bluff State Park. And again, these are all right on the Minnesota-Wisconsin border. So we're making sure that if the spores are in the environment, we're catching them as they come over the border. These are just some pictures of me in the field with my spore traps. I go out every week from Labor Day to like after Halloween, depending on the weather. If it's warmer, like we got last autumn, I usually can go out to the middle of November or so and go out and change the batteries or take all the sampling interfaces off and change them. So I stay pretty busy in the fall. Another picture, you may have actually seen these spore traps out in the environment. This is at William O'Brien State Park, just a little north of the Twin Cities. So after these spore samplers have been out in the environment, how do I quantify and get the spores? So each of these spore samplers have a different collection method. The Burkhardt, the spores are deposited into a microcentrifuge tube or an Eppendorf tube. For the rotorods, the spores are actually on the greased rod or the silicone rod. And then for the windsock, the spores are on the greased petroleum jelly slide. So each of these have different extraction methods, but the extraction methods are functionally the same. We take and we prep the sample and we run it through a DNA processing kit. So wash the DNA and wash all the junk out of there and just get the DNA of the fungal spores that we need. And then we run our samples on quantitative PCR. And what quantitative PCR does is it allows us to detect, one, the presence of Heterobasidion or not, and two, if there is a presence of Heterobasidion, quantify the spores there. And based off how many spores are in the environment, you can make recommendations to the DNR. If they're over a certain threshold, that's indicative of a potential infection or an infection center. So yeah, these infection centers, so the locations that have had high amount of spores in the past 4 years are actively being searched on foot and Google Maps. So you can actually look for circles of death of these conifers along the Highway 61 corridor. And these are areas where we're focusing in our efforts over the next couple of years. And this is also being done with the help of the Department of Natural Resources. And our efforts are being focused near Winona County, not far from the previous Minnesota positive site. And we believe, based on our data, that there's a high probability of infection near Winona County or several infection centers. And that's what we're going to be doing for the next facet of this research. So that was one pathogen that I'm talking about, and I actively research. The next is Phytophthora. So this is totally different research. Heterobasidion sampling occurs in the fall, and Phytophthora detection occurs in the spring and the summer mainly. So maybe you're familiar with Phytophthora, and you're probably familiar with Phytophthora even if you don't know it. Phytophthora was the pathogen that caused the Irish potato famine, Phytophthora infestans. And it also causes sudden oak death, Phytophthora ramorum, which is a very big problem out on the West Coast, and specifically California, Oregon, and up in Vancouver. And a little on the Phytophthora morphology, you don't have to get too bogged down with the figure here. I just want to point out that Phytophthora is an oomycete, so it's a water mold, and it's often miscategorized by a fungus by some people, but it's actually not a fungus. It's closer related to algae than it is to fungi. But it does produce fungal-like structures. So it does have mycelium. It produces a chlamydia spore and these zoospores, which are very important for its life cycle, because what these zoospores do, and you can see here, they have two flagella or little hairs that allow these spores to move through water. And this could be a problem if you have standing water at a nursery or something like that, which I'll touch on in the next slide. And these are what some of the sporangia seen here. This is what they look like underneath the microscope. So Phytophthora. So I probably should back up and say that Minnesota doesn't have a very big growing season, and nurseries around Minnesota ship in a lot of their material from other places around the world and the country that have a better growing season than we do, say Washington, Oregon, Arkansas. And where that becomes a problem is that Phytophoras could be in the samples that are coming over from these states. And they could be on the truckloads of plants and flowers that come and trees. And what we want to do is we want to go to nurseries around the state. And what I do is go to nurseries around the state and actually sample for Phytophthora and making sure that we don't have any scary pathogens in Minnesota. This is just another picture of how large and how many plants these nurseries have at them sometimes. You can just see the fields and fields of plants, and even over past the hoop houses and the grow houses, there's trees on the other side of that. So these are very, very big operations. So when I'm out in the field, what I'm potentially looking for is plants like this arborvitae or cedar that's died here. I would probably sample that entire plant if the nursery let me. I also look for dead and dying plants. So these brown plants like this, I usually focus on rhododendron species because rhododendron are one of the main hosts for Phytophthora. And I also like to sample at these predominantly wet areas around nurseries as well, because like I mentioned before, the zoospores are able to move from either nursery bed to nursery bed or from different places in the nursery to other places in the nursery via water, which could be a problem and is potentially a problem. These are other good candidates of standing water. So I would probably sample the soil in this area here and this area here. And then this would be like a home run for me, because what you can see here are holes of standing water where I assume an arborvitae has been removed because it's either been diseased or it's dying and it's wet. So this is like what I really, really look for and really what I like to see when I go to nurseries. We also have a collaboration with the Minnesota Department of Agriculture, and myself can't sample every nursery around the state, nor do I have access to them being a graduate student. But with our collaboration with the Minnesota Department of Agriculture, what they do is they go and they sample at 35 Christmas tree farms and nurseries across the state. And they do this twice for me. So they collect in May and June. I think that they're just collecting and finishing up their collection for the May-June portion. And they also do a second soil collection around August and September. So I'm really getting a wide breadth of different nurseries I'm sampling and from around the state. So these are what the soil looks like when they come back to the lab. Either I would take these soil samples myself, or they'd be submitted to me via either the Minnesota Department of Ag or the Minnesota Department of Natural Resources. And what I do is I take pears, just au jus pears that you get from the grocery store, and I bait for the Phytophthora. So I take those soil bags and I place pears in deionized water in there. And if the pathogen is present in the soil that I receive or collect, it will actually attack the pear and cause lesions. And I can do a lot of samples at the same time. It's mainly just based off of how many au jus pears and how many good looking au jus pears I can either get from Cub or Hy-Vee, which sometimes becomes a problem. But I'm able to screen a lot of soil in a given year or at any given time if I want to. So this is what a pear would actually look like after 3 to 5 days of being in that soil bag. And this portion here is a squishy portion. It's probably a fungal attack or a fungal infection, and I'm not really interested in this. What I'm interested in is these chocolate brown lesions here seen on your screen. And what I would do is I take a sterile scalpel and tweezers and actually place them into isolation plates. And if you heard Ryan Frankie's talk last month about how he screens for fungi associated with the buckthorn, I do a similar thing, except my media that I'm putting in is specific for the pathogen Phytophthora. So I make isolation plates, and you can see these are growing out. And what I would do is I would take on the leading edge of this one and this one and this one, because these three look unique. and I would place them onto a separate petri dish by themselves and grow out into what we call pure culture. And then from there, I can extract the DNA and get the genus, almost all the time it's Phytophthora, and the species of it, which is significant. So our results from my project over the past 4 years. So today we've processed over 1,500 samples, that's plant and soil samples, and sequenced over 500 Phytophthora from across Minnesota. We found 24 different species of Phytophthora, with 14 being new to the state. And new to the state meaning they have never been detected here. And this is just a graph to show you the frequency of the Phytophthora that I've identified over the course of my project. So the gold bars are the species of Phytophthora that have actually been detected here in Minnesota, either by Brian Schwingle, who did previous research in our lab on it, or based off the literature. So the gold bars are actually the new and the first reports in the state. And you can see all 14 of them there, all the different names of them. So what we want to do and what the future of biosurveillance looks like is first of all, I want to trap beetles for oak wilt. So oak wilt's another invasive pathogen that we want to kind of keep a lid on. And oak wilt, if you know like where Hinckley is and like northern metro up past Asani County, that's where the infection is. And what we're doing is placing spore traps, or not spore traps, we're placing beetle traps out there. And we want to be able to detect the fungus via the beetles that carry the fungus in locations on the leading edge to make sure it's not in counties north of Hinckley. And if it is, we want to make recommendations to the DNR like we do with Heterobasidion. Because this fungus mainly moves not by the beetles, but by the movement of firewood, kind of like Colin will talk about for emerald ash borer. We also want to do pathogenicity studies on the Phytophthora species, the new to the state species, to determine which are most pathogenic. So not a lot's really known about some of these new to the state Phytophthora, and that could be kind of scary. So we want to actually inoculate trees with the Phytophthora that we have in culture and see how much damage they can actually cause. And then long term, and for the rest of my research, I'm going to continue to monitor for Phytophthora and Heterobasidion and other pathogens to help keep the forest healthy. So with that, I would just like to acknowledge my funding agencies, so the Environment and Natural Trust Fund, and also the University of Minnesota Doctoral Dissertation Fellowship. Also, I'd like to acknowledge my advisor, Dr. Robert Blanchette, Benjamin Held, Colin Peters, who's on this call, Andrew Mann, Brian Franke, Eric Otto, my wife Isabel Rickey, Brian Schwingle, who works with the Department of Natural Resources, and Michelle Grabowski, who works with the Minnesota Department of Agriculture, the University of Minnesota Invasive Terrestrial Plants and Pest Center, and Wisconsin Department of Natural Resources. And with that, I would be happy to take any questions people have. Thank you for your attention.
And folks can put their questions in the chat, or feel free to unmute yourself and ask it if it feels easier to do that.
And if you're watching this recorded, feel free to reach out to me via my email and ask me any questions you have as well.
What I first noted, Nick, was how you mentioned that someone had found the red pine fungus in Winona, and how you guys are, looking for stuff. But I think it's just a great opportunity for MMS members. There's lots of us, and we love being out in the woods. So seeing this firsthand from you, and then knowing that we're a lot of eyes and ears and feet in the woods, to kind of keep an eye out and be the citizen scientists that could help you guys kind of find some other locations that you might not make it to because you're only one person.
Yeah, that's actually a great comment, Kathy, and a great observation. I was actually going to end the very end of my presentation before I passed it off to Colin, saying if you do see any of these fruiting bodies, please let me or the Department of Natural Resources know when you're out down in, past Lake City along the 61 corridor, and even north of the cities, William O'Brien or Itasca even. I mean, feel free to always shoot me an email, or if you see something suspicious, because I would more than happy go out and take a look at it or identify it for you.
Awesome. I see Aaron is asking, is there any value in reporting oak wilt to the DNR?
So yes,
He
there
says,
is.
yeah, I believe I've seen it in both the Twin Cities and in Alexandria.
Yep. So yeah, Aaron, that's actually a great question. So there is value in reporting it to the DNR, and it is in the Twin Cities in the North Metro. As far as Alexandria, I believe the DNR do know that it's in Alexandria, Minnesota, and that's kind of like on the leading edge of the 94 corridor where it's found. But yeah, what we're looking for, and if you see it past there, is the places just a little north of there, on the leading edge of infection.
Let's see, Nick, can you hear me?
Yep.
Okay. Yeah, Ron here.
Hey, Ron.
On our first foray of the season, it was actually in was at Battle Creek Park. On Red Pond there, we found Cryptoporus vulvatus, I think.
Yeah.
Anyway, I had never found that before in Minnesota.
Mhm.
And I read something that that's a pine bark beetle that introduces it, and is that significant?
So it is significant that it's vectored by the pine bark beetle. And yeah, it's pretty uncommon in the Twin Cities, but where we see more of it and most of it, it's up in like the Cloquet area and in northern Minnesota where we have more of those pine plantations. So it would be a little odd to see it down here, but I wouldn't rule it out. As far as tree health wise, it does cause a little bit of damage to the tree, but I mean, because it is a bark beetle and a fungus, but nothing too scary to be worried about there.
Okay. Yeah, I was just curious. Yeah.
Mhm.
Thanks.
Yeah, it's a good find.
Do you mind answering a couple more questions about oak wilt? So I have
sure.
a lot of, and it's just all hearsay. I've heard things that it's like oak wilt, and I live in Forest Lake, so I feel like it's sort of everywhere there. And I happen to have a lot of oaks in my yard, and people say that if a red oak gets oak wilt, it dies from it faster than a white oak.
Mhm.
Is that true?
Yep, so that's true. So both of the oaks in the red oak family, so the pin oaks as well, reds and pins will die within the year. But if it's a white oak, you can actually treat it sometimes with fungicides therapeutically, and just the white oaks take a longer to die, and some of them can actually persist and hang on for decades, and if not, yeah, 10 years, 15 years, depending on infection. Yeah, with that, I would thank everyone for their attention, and yeah, I'll pass it off to Colin Peters.
Awesome. Thanks so much. Really appreciate it.
Yeah, thank you guys.
And before Colin shares his screen, I'm going to do a quick introduction. I forgot to mention that our graduate scholarship program was started in memory of James Swanson and Doris Johannes, who were longtime members. And both passed away within the same couple years, and we wanted to find a way to honor them. So it's great to have people doing research and getting a little funding in their name. And Colin, thank you for being here tonight. I heard that you were just at the lab working late. So we appreciate you taking a break to tell us about it. And Colin is also doing a lecture on native entomopathogenic fungi, which are insect pathogens that are found to be associated with the emerald ash borer in Minnesota. Colin is a second-year master's student at the University of Minnesota, where he's also advised by Dr. Robert Blanchette. He received a bachelor's in plant science from the U of M in 2019. And after completing his undergrad, he spent a year in the Conservation Corps and worked in tree care before returning to school. So he is an avid forager of wild mushrooms and has been a member of MMS since 2023.
Yeah, thank you, Kathy. All right. So yeah, my name is Colin. Tonight, I'm going to be sharing with you some very important work that we're doing, trying to develop a biological control for the emerald ash borer using native entomopathogenic fungi. Kathy did a great job, but I'll start by just mentioning a little bit about myself. I received my bachelor's in science in plant science from the University of Minnesota in 2019. After which point I spent a few years in the tree care industry. I was a foreman of tree injections, as well as working in the conservation industry at various jobs. So I've returned, let's see, last January to school at the University of Minnesota to start my master's program with Professor Robert Blanchette. And I'm on my second year now. Fingers crossed, I can transition to the PhD track real soon here. So tonight, I'm going to give a brief background on emerald ash borer, or EAB, and the devastation that it has caused across Minnesota and North America, as well as the fungi that have been described to be associated with it. Some of that work was done by Nick Rajtar. And from there, I'll touch on the work we're doing now to characterize the virulence or pathogenicity of these entomopathogenic fungi that are capable of infecting the insect and are potentially useful as a biocontrol. And finally, I'll touch on two field application methods that we are investigating for deployment across the state to slow down the spread of this beetle and to prolong the life of ash trees. So let's dive in. The emerald ash borer, or EAB, as I'll refer to it throughout the presentation, Agrylus planipennis. I am very confident that most of us on this call have some connection to this invasive beetle, and particularly with the destruction to trees that it can cause. It's native to eastern China and the Koreas, as well as Siberia, and was first detected in North America in 2002 near Detroit, Michigan. But it's believed that the beetle actually arrived in the late 1990s via infested wood shipping material. Since its discovery in 2002, it is spreading across North America. It was detected first in Minnesota, in St. Paul, in 2009. And the significance here is that all of the species of ash that are native to North America are very susceptible to this beetle. And so we're at risk of losing this entire genus, really, across the continent. Before I start talking more, I'd like to just cover some of the life cycle of emerald ash borer in the context of Minnesota here. So in Minnesota, the eggs are laying in the month of June, and they are laid in the bark crevices or underneath bark plates on the branches and trunks of ash trees. And these eggs will hatch in the month of July. And that's when a larva, seen in the middle there, will tunnel in. It'll chew its way in beneath the bark of the tree, and it feeds on the vascular tissue within that tree, particularly the phloem. And it'll serpentine around, carving out these galleries, we call them. And then there's also the adult beetles, or let me back up a second. The larvae will feed throughout the summer and into the fall. And when winter comes, they will chew out this pupation chamber just underneath the bark in which they'll overwinter. And the following spring, they'll complete pupation and emerge from the bark as adult beetles. And these adult beetles will briefly feed on some of the ash foliage, and they'll find one another and mate to lay eggs and restart the cycle. And while the adults do feed on the leaves, it's generally insignificant to the tree's health, not very detrimental. But the real damage that's done by this invasive insect is from the larval stage, because these larvae will create these galleries and tunnel around in the vascular tissue, severing the conductive pathways inside the tree that connect the roots to the canopy. So that's what is killing the trees. Oftentimes when EAB will first infest a tree, it goes largely undetected for the first 1 to 2 years. The tree will seem totally healthy. Some of the first symptoms you might start to see after those couple of years are woodpecker holes, which are shown in the picture here. This is caused by woodpeckers searching for the EAB larvae underneath the bark because they forage for them as food. And so they'll strip away the outer bark and make holes in the tree. You can start to see the canopy thin. It'll produce fewer leaves, and the leaves it does produce are smaller and lighter green in color. And portions of the canopy will even start to die back completely and not produce leaves. You'll start to see these bark splits, which are caused as the tree starts to form callus tissue, trying to heal over these larval galleries. That callus tissue will push up and exert pressure on the bark, which will then split open, revealing the galleries beneath. There are characteristic D-shaped exit holes that you can look for. And these are formed by the adult beetles that chew their way out through the tree. If you peel back the bark beneath some of these, the woodpecker holes or the D-shaped exit holes, you'll find characteristic serpentine larval galleries filled with frass. And in the late stages of infestation, the canopy will die back completely, and a stress response from the tree as it's trying its best to photosynthesize, it will put out this new growth, these epicormic sprouts, which are formed near and around the base of the tree. So since its introduction to Minnesota in 2009 in St. Paul, EAB has rapidly spread. And according to the Minnesota Department of Agriculture, it has now been confirmed in 52 Minnesota counties. And you can see here the generally infested areas are outlined in this lime green around the state, with the main infestation pocket emanating from the Twin Cities. But there are a couple significant pockets, especially this one up by Duluth, as well as one just beginning over in Moorhead and by Fargo. And these are, this is due to people moving infested firewood. They'll cut down an ash tree, say, and they aren't aware that this invasive insect is just waiting to emerge beneath the surface of the bark. They'll move that to a new location outside of quarantine boundaries. And come spring, these beetles are just going to do what they do. And so we get these pockets of infestation popping up long distances from where they might be expected. So Minnesota is home to an estimated 1 billion ash trees. And a large amount of those are in the northern portions of the state in black ash swamps. This is a huge, immense natural resource, and it's crucial that we do something now to slow down the spread of this beetle so that we can transition more smoothly, so that our forests can transition more smoothly as this beetle inevitably moves across the state. So as this beetle has been introduced to North America and has spread, it is quite literally carving a new niche in our local environments. And a previous study with both Nick Rajtar and Robert Blanchette, who are on the call, looked at the fungi that are associated with these larval galleries. And they took wood samples from the larval galleries from across Minnesota, and then they grew out any fungi in culture, and they extracted the DNA from these fungi and sequenced them to identify them. And they found a huge diversity of different species that are present in these larval galleries. And they went further to characterize essentially the lifestyles of these different groups of fungi, or what we call functional guilds. So that what they found were three main groups, with the most frequently isolated functional guild being canker pathogens, which Nick Rajtar had worked on for his master's work, and found that indeed there are some of these canker pathogens, canker fungi, that the beetle is bringing in that contribute to the decline of ash trees, in addition to the just actual mechanical damage caused by the insect feeding. These canker fungi are also working at killing this tree. There are wood decay fungi present, and then there are also the entomopathogens, or insect killers. And tonight we're going to be focusing on the entomopathogen group, and of which I've listed the most frequently isolated genera from that study. And so for my work, I have inherited this collection of entomopathogens, and I have two broad goals for the project. The first being to characterize the virulence of these native entomopathogens against the beetle, the emerald ash borer beetle, and further to test field application methods with the most promising fungi. And so, yeah, I'll touch on how I do this. So first, talking about characterizing the virulence of these fungi, we do this using virulence assays. And so essentially what I do is I go out in early March, and I harvest an infested ash tree and cut it into 24-inch log segments, and then place those logs inside these rearing tubes pictured here. And after about 3 weeks in these logs, EAB beetles will start to emerge from the logs. And the beetles are highly phototropic. They love the sunlight. And so they'll actually kind of collect themselves in these clear cups that are attached to the log, or to the rearing tubes. And then I can go in and just unscrew the cup and put a lid on it. And then I have beetles to work with. And so here are some pictured. And I can get, from these 9 logs at a time, I can get hundreds of beetles to flush out of them. And I think you can see that in this picture. There are quite a few. From there, we keep them in mesh cages in the greenhouse, and we provide them with fresh ash leaves, which keeps them happy. So with those beetles, I'll then take media plates of these entomopathogenic fungi, and I'll harvest spores from these PDA media plates. And so I put this tween and water solution onto the plates and kind of gently scrape them, and then filter that through cheesecloth before using a hemocytometer to then adjust those solutions to a standard concentration. And for those of you who aren't familiar, a hemocytometer is originally used to count the concentration of blood cells in serum, so that's where the name comes from. But essentially, it's a means of counting the concentration of spores in a solution using this laser-etched microscope slide. And this is kind of the final product once they've been adjusted to that standard concentration. And so they're ready to have beetles dipped into them there. Once the beetles have been dipped into the solution, we incubate them in a growth chamber and provide leaves for them. And you can see here, as far as an incubation chamber, we took just takeout containers that you might get from any restaurant, right? And we cut holes in them with cheesecloth mesh to provide ventilation for the beetles. And then we punched a hole in one side, and we have the ash leaf there with the petiole sticking into a falcon tube filled with water so that it stays fresh for, it lasts about 2 days, and then you have to replace it. But the beetles are kept in there, and we check them daily for mortality. And then any that have died, we surface sterilize, and we put into a humid chamber to encourage mycosis, to encourage the fungus that is potentially inside of the beetle and has killed it to sprout out from the beetle and produce spores. And with this method, we found that there are some fungi that show preliminary promise in their ability to infect the beetle. And so pictured here is an isolate of Purpuricillium lilacinum sprouting out from a dead EAB cadaver in the lab. So this year we're continuing our virulence assays, but using different inoculation methods that are more transferable directly to the field. So one is a fungal pouch walk, and that's pictured on the right here. This picture is from a preliminary trial that I ran using tenebrio or darkling beetles. These are the beetles that mealworms turn into. And they're real easy to work with in the lab because they can't fly. But essentially, we make up these fungal pouches, which are just colonized grain that is housed in a cheesecloth pouch within an embroidery hoop. And we drop the beetles onto them and let them walk around and get coated in fungal material. And then we're also trying a pre-emergent trunk spray. So we have infested logs, and we will spray them with a spore solution so that as the adult beetles emerge, they have to chew through that bark and consume the fungal spore solution. And some entomopathogenic fungi, such as Bovaria bassiana, are indeed capable of infecting insects through the gut tract. So this could be a promising means of biocontrol. So moving on to field application methods, I'll talk first about these auto-dissemination devices, or ADDs, as I'll refer to them. ADDs are Lindgren funnel traps, which are pictured up here in the upper right. It's essentially a series of plastic funnels that is hung up in the canopy of an ash tree. And they are baited with chemical lures that are attractive to EAB. One is an EAB-specific sex pheromone called 3Z-lactone. And then one is a 3Z-hexanol, which is a tree volatile that ash trees will give off when they're stressed or under attack. And both of these are attractive to EAB. So the EAB come to the trap. They try to land on any of these funnels, but the funnels are coated in a non-stick polymer called fluon, and they fall all the way down into this collection cup, which contains a living culture of fungus. It contains one of those fungal pouches that I had pictured or shown before. And then there are exit holes in that collection cup so that the beetles, once coated in spores, can escape, and they'll spread the fungus to one another through mating. So last summer in Minnetonka, along the 494 corridor, we installed 16 of these ADDs. And you can see the ADD pictured there up in the tree's canopy. And we paired each one of those ADDs with a sticky panel trap, which is coated in a glue and is also attractive to EAB, so that we are both trying to infect the emerald ash borer and then capture them for analysis in the lab. And we used a fluorescent pigment powder within that collection cup on the ADD. And that was a means of visually indicating which beetles had visited or interacted with our trap. And we visited the site for 4 weeks, weekly, collecting all the beetles from these sticky panel traps. So these are the fungal pouches that I was mentioning. And here's an example of some emerald ash borer caught on the sticky panel traps. This is actually, these two pictures are the same beetle, which is right here. This picture on the left is just normal light, and then the picture on the right is me shining a UV flashlight at the trap. And you can see this orange fluorescence on the abdomen. That is due to our fluorescent pigment powder. And so we know that this beetle interacted with our trap. Once we collect them from the field, we surface sterilize them so that, and then keep them in a humid chamber so that any fungi that is inside the beetle will come out. And you can see here that, again, this is normal light, whereas this is under UV. And you can see the fluorescence on the abdomen, in addition to these signs of infection from our biocontrol fungus. And so this year, we want to continue that work. We have set up plots along the advancing front of emerald ash borer across the state. So we have plots at the Minnesota Landscape Arboretum, Crown College, which is out west of the metro, Interstate, Wild River, and Jay Cook State Park, and then a wildlife management area called Spectacle Lake, northwest of the cities. And we have a treatment and a control plot at each site, and we're going to be measuring the EAB population growth rates across years there, as well as the rate of canopy decline in the ash trees. to see if these traps can prolong the life of these ash trees. Okay, and finally, I want to talk about another field application method we're investigating, the injections with fungi. So many of these entomopathogenic fungi, whereas we might think of fungi as mycelium, these networks of hyphae, or we might think of fungi as yeasts, these single cells that can bud and reproduce very quickly. Many of these entomopathogens are dimorphic, or they're capable of both types of growth. And what determines which type of growth they produce are the environmental conditions that they find themselves in. So in nature, these entomopathogens, when they're inside an insect, will produce this budding yeast stage, which we call blastospores. Whereas once they're outside of an insect, they would produce true hyphae. And the spores they would produce are conidia. And what's important here is that conidia are very hydrophobic. And so if you're trying to make a solution with them, it's harder to keep those conidia suspended in solution. Whereas blastospores are less hydrophobic. And so it's easier to make a solution of them. And so what we did is we encouraged the production of blastospores with an isolate of Bovaria bassiana by growing it in liquid culture at a very high RPM on this incubator shaker. And after about a week of growth like that, the liquid cultures were filtered through a Buchner funnel, pictured on the right, to remove any sediments or mycelial fragments that might clog up the vessels in a tree. The filtered product was then centrifuged, and the supernatant, or the liquid on top, was poured off so that the resulting product is this pellet of blastospores at the bottom of the tube, which can then be dried and stored for later use. And once you take that into the field, you would add water to rehydrate and resuspend that pellet. And under the microscope, this is the product you get. This generally uniform solution of blastospores. And so we use a process that's very similar to how fungicide injections are done for oak wilt treatments. Using this macro infusion manual pump kit, we excavate around the root flares of the trees, and using a hand drill, we drill in every 3 inches around the root flare and insert these tubes and tees into the tree. The inoculum solution goes into the tank, and we pressurize it, and then the tree will take it up within, we've seen anywhere from a half an hour to 2 hours, depending on the environmental conditions and humidity, sunlight, etc. So last year in Minnetonka, we injected 12 trees, each with 1 liter of inoculum, and 3 treatments. One was water. Then there was one with what we call the low concentration treatment, and then a treatment that was the high concentration. And what we wanted to do is just see, is our fungus distributing throughout the tree? Is it actually getting into the tree and effectively up into the canopy where it's most important to, in terms of protecting from emerald ash borer? If the fungus truly can become established in the tree from this injection, there is potential that as the emerald ash borer larvae start to feed they would ingest that fungus, and this could potentially be a means of biocontrol. So once we injected the trees, we returned at 3 weeks post-injection, 5 weeks, and then 15 weeks post-injection. We took increment borer samples at the 3 and 5 week marks. And then at the 15-week mark, we harvested the trees and took cookies from various heights up the main stem of the tree as well as in the canopy. And then we took those wood samples and plated them onto semi-selective SDA media. And any colonies of fungi that grew out that looked like Bovaria were then moved to pure culture on PDA plates and were confirmed using microscopy to be Bovaria. And we were seeing that we did not see any Bovaria come back from the controls or the low concentration treatment wood samples. But in 11 of the 52 wood samples from the high concentration treatment, we had Bovaria bassiana present. And so this seems promising. And with this year, we want to try more injections. And just last week, we went out and injected at the Sand Dunes State Forest. And we've completed 16 trees. And this time, the trees are receiving twice the volume and or over 3 times the concentration of spores that they had received last year. And we will continue to do the culture-based semi-selective media confirmation, as well as molecular confirmation by extracting DNA from the wood and running PCR on it. This is acknowledgement of funding for the project from the Minnesota Invasive Terrestrial Plants and Pest Center and the Environment and Natural Resources Trust Fund. We have several collaborators without whom this project wouldn't be possible. City of Minnetonka, the Minnesota DNR, the Landscape Arboretum, and Crown College. And I want to personally thank Robert Blanchette, Benjamin Held, Nick Rajtar, Sophia Semeto, Ryan Franke, and Andrew Mann for all of the help and guidance that you have provided me along the way. And with that, I'm happy to take any questions.
Wow, thank you so much. Colin and Nick, I'll put your email in the chat for later. I'm looking at the chat here. Alicia, are other insects attracted to the ADDs? And does the fungus have an effect on the ash tree?
Yeah, that's great questions. So we have found some insects inside these ADDs that aren't emerald ash borer. We are trying to target EAB specifically by using that EAB specific sex pheromone and minimize the number of non-target insects that are interacting with that trap. But indeed, we have found other insects in there. And let's see, the other question was, how is the fungus affecting the trees? So based on last year's injections, when we harvested the trees, they were comparing them to the controls. The canopies were similar. And also we didn't note any indications of decay or degradation in the sapwood in the injected trees. And so these fungi are in there and not causing harm to the ash trees.
Cool. I'm just curious how big does an ash tree have to be attacked? It's like, does it have to get to a certain size? I think to me, it seems like as I see a lot of the ash trees in my yard dying, I don't know if a lot of things when they're sort of on the way out, they drop a lot of seeds. So then I have a lot of small ash trees that grow in my garden. And then I feel bad pulling them like I used to as weeds because I'm like, oh, what if you're one that could live? And they seem like maybe they're just too little to be attacked. I don't know if, they need to be like 2 years old or 3 years old. And if there's any point in not pulling them out.
I've seen that the larger ash trees are attacked first. And even in heavily infested sites, there can be young seedlings that are still alive. This is because the larvae actually, they need a certain threshold of vascular tissue to feed on, right, and develop. And so these smaller trees that are about 2 inches or less in diameter, you'll find are typically less favored by the beetle.
Cool, thank you. Let's see. And then Alicia's asking a follow-up question. Will there be longer studies to see the effect of the fungus over time?
Yeah. So Alicia, are you asking about the injections or the auto-dissemination traps? Because we have initiated a 3-year study on the auto-dissemination traps with those 6 locations along the advancing front, where we're going to be comparing how quickly the trees are dying and how quickly the emerald ash borer populations are exploding. And we want to initiate essentially a similar study using injections once we have a reliable protocol established for that.
Alicia, do you have any other notes or questions? You're welcome to unmute if you like. There's also a question from Howard for Colin and Nick. Maybe Nick had to head out. Do you ever have time to notice other fungi in your time in the woods?
Oh, that's a fantastic question.
Besides that giant puffball.
Yeah, I guess that kind of answers it, right?
Did you steal Nick's pillow?
May have.
That's great. I am always looking for interesting fungi. That is one of the great joys in life, is it not?
Yeah, one of the perks of field work too is like going and looking for edible fungi while you're out in these forest stands at every different time of year.
Mm-hmm.
Nice. Any other questions for Colin or Nick? I did just paste both of your emails in the chat in case people find it easier to cut and paste to save those. Make sure you got them right. So Colin, is the thought that, so it's like if the fungi is hazardous to the beetles when they've eaten it, then when they kind of emerge, the fungus would grow on them before they could lay their eggs, or maybe they don't even get to emerge because they're in their little space in the tree and the fungus attacks them before they're even able to leave.
Right. So in the case of a spray, like a pre-emergent spray, That would be the goal would be that the adults, as they're chewing through that outer layer of bark, are ingesting the fungus, and that it would kill them before they're able to lay eggs. We don't have data on that yet, but we are hoping this summer to start those pre-emergent log spray assays in the lab.
Very cool.
And if anybody has additional questions, I'm totally happy to take emails.
Great. Yeah, if anyone has anything and they just want to unmute and ask in person, or any other related questions, any other unrelated questions or things to share. Awesome. Yeah, this is really exciting. It's really fun to see how much really important studies are being done just kind of in our backyard. And, you know, even just seeing those maps that you showed of how much stuff is spread, it's sort of daunting. It's a bummer to see. I know. So I live again just north of the cities and thought, oh, maybe it won't come here. And now I've kind of seen all my ash trees die.
Mm-hmm. Yeah, I feel like more and more everybody has a story of losing an ash tree, right?
So is it better if you do lose a decent sized ash tree? Is it better to cut it down and try to dispose of it before the beetles, you know, could emerge and spread more? Does it actually help, or have they already moved on once your tree is dead and it's too late? Or, I mean, is there?
Once the tree is dead, you know, there have been multiple generations that have already emerged from that tree, and oftentimes by the point that the tree is fully dead, the beetles will have mostly moved on. So if you catch an infestation in your tree early, and you know, there are options like insecticide injections that are extremely effective. But if you're not inclined to go down that route, removing the tree before it dies and becomes this hazardous, decaying, just hazard, that's the best case scenario in terms of reducing the impact that tree will have on the EAB population growth, right? Yeah, so I'd say catching the infestation early is the key.
Well, and Sheila noted, well, first, Copper has noted that it's really cool to see biologics being used as potential control. And Stina noted, we've lost all of our ash and elm trees on our property. There's a few saplings remaining, and I hope they can last. So glad you guys are doing this work.
Appreciate that.
All right. Well, thank you so much. Yeah, it's really, really great to hear what's going on at the U.